Hi everyone! You may have heard of genetic conditions such as sickle cell disease and alpha thalassemia before, and learned that they are all disorders of the blood. But have you ever thought about how they are different and what really sets them apart? Hemoglobinopathies is what we call a fairly large group of inherited blood disorders that involve disruption of hemoglobin. To really understand the basis of all these conditions, we have to first look at this hemoglobin protein, whose main job is to carry oxygen. This protein is abundantly found in red blood cells, and it is also commonly used in biology class as an example of a quaternary structure, because it is made up of smaller protein subunits. Zooming in, we see that there are 4 protein subunits: 2 alpha chains and 2 beta chains. There are also heme groups, which are components containing iron that actually help to hold on to the oxygen, but these will not be our main focus in this video. The alpha chains, also known as alpha globins, are proteins encoded by both the HbA1 and HbA2 genes. These genes are almost exactly the same, and they are both located on chromosome 16 within close proximity to each other. It doesn't matter which gene the proteins are encoded from. You can think of them as all going into the same pile in a hemoglobin-making factory. On the other hand, the beta chains, or beta globins, are proteins that are encoded by the HBB gene. There is only one HBB gene, and it's located on chromosome 11. There are 2 major categories of hemoglobinopathies, defined by the 2 major ways in which things can go wrong when making globins. The first is when a genetic change causes there to be less globins being produced. These are called thalassemia syndromes, or you can think of them as quantitative defects. The second way in which things can go wrong is when a genetic change affects the structure of the globins being produced. There might still be the same amount that is being made, but there is something in the structure itself that is altered. These are structural hemoglobin variants, or qualitative defects. Nevertheless, for both types of hemoglobinopathies, they are usually inherited in an autosomal recessive manner. Let's take a closer look at thalassemias first, which occur from genetic changes that lead to a decreased quantity in the number of globin chains being produced. Alpha thalassemia is one example of a genetic condition that is in this category, and it is marked by a reduction in the production of alpha globin chains. Remember when we talked about how there are 2 genes that are involved in making the alpha globins, HbA1 and HbA2. Each gene has 2 copies, so altogether there are 4 alpha globin making stations. The most common type of genetic change that lead to alpha thalassemia are gene deletions, and the severity of symptoms depend on how many copies are deleted. People with 1 gene copy deleted often experience no symptoms, and for this reason, they are commonly referred to as silent carriers. On the opposite end of the spectrum, there is alpha thalassemia major, where both copies of HbA1 and HbA2 are deleted, and there is no production of alpha globin chains at all. In this case, the hemoglobin being formed is an abnormal variant called hemoglobin Bart's, and people with this condition die shortly after birth or are stillborn. Let's now take a look at an example of a structural hemoglobinopathy: sickle cell disease. Again, This occurs when genetic changes alter the physical structure of the hemoglobin protein being made. In sickle cell disease, a specific mutation in the gene HBB changes the biochemical properties of the amino acid chain, and this causes the resulting hemoglobin to form a characteristic sickle shape and clump together in blood vessels. Typical of an autosomal recessive inheritance, having this mutation in one copy of the HBB gene usually do not lead to any symptoms. But when an individual inherits two HBB copies with the variant, they will have sickle cell disease with accompanying symptoms of pain, organ damage, and anemia, or lowered levels of red blood cells. You may be wondering why sickle cell disease would be associated with anemia if it's classified as a structural hemoglobinopathy. The same amount of globin and therefore hemoglobin should be made, right? So the amount of red blood cells carrying oxygen shouldn't change. Well, people with sickle cell disease experience a type of anemia called hemolytic anemia, where the sickling effect significantly shortens the lifespan of red blood cells so that they are damaged faster than they can be replaced. To summarize, there are several key facts to know about hemoglobinopathies. First of all, hemoglobin is a protein that is made up of 2 alpha chains and 2 beta chains. Now, keep in mind, this is usually the case for adults. If you want to learn more about other types of hemoglobin, you can watch my other video here. Secondly, these different components of hemoglobin are encoded by different genes. The alpha chains are either encoded by HBA1 or HBA2. The beta chains are encoded by the HBB gene instead. Thirdly, there are 2 major types of genetic variation that can occur: quantitative and qualitative. Quantitative hemoglobinopathies occur when less alpha or beta chain is produced. Examples include alpha thalassemia and beta thalassemia. Qualitative hemoglobinopathies occur when a specific genetic variant causes a significant change in the biochemical properties of the resulting globin chain. An example of this is sickle cell disease.